Hi, my name is Samara Davis, and today I'm presenting my research environmental DNA analysis to determine population characteristics of elusive ephemeral pool-breeding mole salamanders in relation to the effects of climate change. Currently, 41% of amphibian species are threatened with extinction according to the IUCN Red List. In comparison to other animal classes, amphibians are exhibiting the most extreme population declines and range reductions due to many biological and physiological factors, including habitat fragmentation, climate shifts, invasive species, and infectious disease. One habitat that many amphibians are dependent on are ephemeral pools, which are temporary wetlands that fill up in the spring and dry out in the summer due to multiple inflows of water shown in this diagram. Since there is no single inflow of water, predatory fish cannot survive in this environment, making it very important for amphibian reproduction. Vernal pools and woodland pools are two types of ephemeral pools and terms that I use interchangeably. Two vernal pool breeding salamanders include Ambosona jeffersonianum and Ambosona laterale. I chose these as the target species for this study as they are listed as special concern by the New York State DEC because of their declining population. They're considered harbingers of climate change because of their sensitivity to environmental shifts, and they are naturally sedentary and elusive, which makes them difficult to traditionally biomonitor. One monitoring technique that overcomes these challenges is environmental DNA analysis. eDNA metabarcoding is a type of eDNA analysis that simultaneously determines the presence of multiple species in an environmental sample like soil, permafrost, or fresh water. An eDNA detection in a freshwater sample would indicate that the species was present in the area in the past 1 to 2 weeks. One amphibian study used a broad-scale demographic approach to compare climate drivers to amphibian population responses in an effort to gauge the effect of climate change on amphibians. This produced varied results that ultimately could not contribute to conservation, so Mutz et al. Concluded that to determine the effect of climate change on amphibians, a fine-scale approach must be applied, which is integrated into my study. One ephemeral pool study found that climate change resulted in shorter hydro periods and longer dry periods, which caused less successful reproduction of pool breeding amphibians. Brooks then concluded that vernal pool habitats are increasingly threatened as climate change progresses. Another amphibian study by Lipovsky et al. Compared physiological risks like evaporative water loss and critical temperature thresholds with climate projections. It was concluded that climate change severely threatens amphibians at an increasing rate, supporting the notion that amphibians are particularly vulnerable to the effects of climate change. Previous research has yet to apply a passive biomonitoring technique to elusive mole salamander species. The purpose of my study was to detect two threatened pool breeding amphibian species using environmental DNA analysis, as well as to compare historical data to recent environmental data on vernal pools. I hypothesized that eDNA analysis would reveal the presence of both the elusive Jefferson and blue-spotted salamanders, as well as wood frogs as a positive control. I also predicted that historical data will show a climate shift and in turn an increased threat to vernal pools. The first step in my study was finding areas within the lower Hudson Valley region with access to vernal and woodland pools. I used the New York State DEC's Environmental Resource Mapper and Google Maps to visually identify forested wetlands, shown by the dark green. I then acquired sampling permissions and a Westchester County Park special use permit to access these wetlands. The target species in this study, including Ambosona jeffersonianum and Ambosona laterale, was Rana sylvatica as a positive control to ensure that DNA did not degrade during storage. This dataset included both known vernal pool vertebrates as well as any species identified in the field. I collected environmental data at the time of sampling for areas like air temperature, water temperature, and visual identification of my main target species. I then compared this environmental data to historical data from various citizen science initiatives, including iNaturalist and the Amphibian Migrations and Road Crossings Project, of which I personally participated in. I collected 500 milliliter water samples from bodies of water, ideally isolated breeding pools, but other wetlands were included to get a more comprehensive idea of the species demographics. These samples were then frozen until they can be analyzed, and I collected a negative control of spring water on each sampling day to ensure there was no DNA contamination. I thawed and filtered the samples through 0.45 micron filters with the suction equipment shown. These were then frozen and partially dried until they could be sent to the lab where they were purified with a buffer and DNA was extracted. From the extracted DNA, the 12S mitochondrial rRNA gene was amplified through a polymerase chain reaction. It was then sequenced through next-generation sequencing with Amplicon EZ at GeneWiz for the primer IT, AFAR, forward and reverse primers. The reference sequences for vernal pool vertebrates were obtained through GenBank accessed through NCBI, and I used MegaX to align and organize the sequence data with the muscle alignment software shown here. The sample sequences were put through the DADA2 bioinformatics software with screens for errors, pairs forward and reverse reads, and counts the number of reads per sequence. These sequences were then blasted against GenBank nucleotide data, and I considered a detection any alignment with over 99% identity. My preliminary results have shown DNA yields for samples 1.3 and sample 3.2, which sample 1.3 contained 0.837 nanograms of DNA per microliter. In comparison, sample 3.2 contained 1.1 nanograms of DNA per microliter, and the difference is visually evident in this illuminated diagram. Sample 1.3 had less DNA because it was collected before breeding season, and once sequenced, it was negative for all vernal pool vertebrates. In comparison, sample 3.2 was collected at the peak of breeding season and contained 13,484 sequence reads for Rana sylvatica with 100% identity, 48,711 sequence reads for an Ambistoma genus with 97.8% identity, and 46 sequence reads for Ambistoma maculatum with 96.5% identity. Some more DNA yields were collected for sample 2.0, which was a negative control and contained 0 nanograms of DNA per microliter, showing that there was no DNA contamination in this part of the process on that sampling day. In comparison, sample 5.5 contained 18 nanograms of DNA per microliter, which correlates with the activity in these pools during the time it was collected. I hypothesized that eDNA analysis would reveal the presence of both the elusive Jefferson and blue-spotted salamanders, as well as wood frogs. My preliminary results have revealed the presence of a mole salamander species and a detection of wood frog species. Some limitations of this research approach include the possibility for contamination between samples, as many are collected on the same day with the same equipment. There is also the risk of DNA degradation during storage, as they are frozen for some time before they are thawed and filtered, an incomplete reference database, as there is no reference sequence for the Ambistoma jeffersonianum, and a short data collection window, but this is mostly avoided because I supplement the study with historical data. In the future, I plan to compile and compare environmental data to historical data to show a climate shift. I also plan to filter and analyze the remaining samples I have and purify and extract that DNA at lab over the summer. This research contributes to the understanding of the effect of climate change on vernal pools and the population status of threatened mole salamanders, which both support conservation planning. An eDNA analysis of elusive amphibian species contributes to increased and more effective biomonitoring, which also supports conservation planning. From my preliminary results, I can conclude that environmental DNA analysis effectively detects pool breeding amphibians in a threatened ephemeral pool habitat. I would like to thank my mentor for providing excellent guidance and supplies for this project, my friends and family for all their encouragement, and my science research teachers for providing endless support and resources. Thank you for your time, and I can't answer questions, so.